Chapter 11.
Oh, yeah. Oh, God.
We're making it. So are we making it?
Oh, I put 2019.
Oh, cool. I mean,
I'd be in high school. Senior year of high school.
Not the worst year. All right, so chapter 11 is dealing with meiosis, right? So we talked about, when was that? Is that unit 2 when we did mitosis? Yeah, I guess it must have been. So like a million years ago, we talked about mitosis. Meiosis is similar. So it won't be like 100% new. Pieces of it will look, it'll be words and things that you've heard before. But it's kind of like a special case of cell division that serves a special purpose, specifically sexual reproduction, right? In sexually reproducing organisms, we need something like meiosis. So chapter 11, we're going to talk about why and what's happening. And then kind of like we did with mitosis, kind of walk through the actual process. So, right. So the first chunk is that this idea that sexual reproduction requires meiosis. So we're going to talk about why and talk about kind of ending and talk about the fact that as a result of that, we've got these kind of different sorts of cells in our body, the two different kinds of cell division. There we go. So sexual reproduction, general concept, a new individual is produced as a result of the genetic contribution of two cells, right? That makes sense. Right. And the two cells in question are an egg and a sperm cell, right? These are gametes, sexual reproduction cells, things we're vaguely familiar with. And so these two cells come together in sexual reproduction, right? All the other cells in your body are somatic cells. They are non-reproductive, right? The cells in your arm or in your ear are not reproductive. They are somatic, or body is kind of the root of that word. They are somatic cells, right? And so subsequently, those somatic cells, and we'll hit this again later, are not capable of undergoing meiosis. So we've got gametes and we've got somatic cells. So when a sperm and egg cell fuse to each other, they form a single cell. So it's not like a sperm and an egg come together and you get these two cells that are stuck together. They literally become a single cell, right? And so that fusion is called fertilization or syngamy, right? The gametes are coming together, it's kind of the root of that word, right? And so we'll kind of walk through all of this, right? But your haploid sperm and your haploid egg, right? So they're each only carrying kind of half a set of DNA. So when they fuse together, the resultant diploid cell, right, has two copies of everything, right? A copy of everything from the mother and a copy of everything from the father. Now, for this to work, something has to be reducing chromosome number, right? Because each of your cells, right, if we just look at any of our cells, you have 46 chromosomes in the nucleus of every somatic cell that you have. If we just use these somatic cells to do reproduction, we're combining a 46 chromosome cell with a 46 chromosome cell and getting a 92 chromosome cell. I'm not going to do any more math after that, but you'd be 4N and then you'd be 8N, right? And then 16 and then 32, and the cells would just get enormous and ridiculous and be crazy. So something has to happen to reduce the number of chromosomes in the cell for reproduction to kind of happen in a sane sort of way, right? So meiosis, this is where meiosis comes in. So meiosis is the kind of cell division that leads to sexual reproduction, and it's a special reductive division. So it is a cell division that will serve to take cells from diploid, right? We're diploid organisms. We have two copies of all 23 of our chromosomes in every cell, to a haploid cell, which only has one copy of each of those 23 chromosomes, right? So that every sperm cell carries 23 chromosomes, every egg cell carries 23 chromosomes. And so that the offspring that's created has the appropriate number, 46 of chromosomes, right? And so this kind of cycle of meiosis, creating haploid cells, and then fertilization, creating a diploid cell, is a full reproductive cycle in that sense, right? So sexual reproduction is this process ongoing, right, through species and evolution, right? Of alternating between meiosis to create haploid cells, and then using those haploid cells to undergo fertilization, which creates a diploid cell. And back and forth, and back and forth, right? So, all sexually reproducing organisms are alternating between haploid and diploid chromosome numbers, essentially. And then in animals, kind of where we'll keep our focus, animals, the zygote undergoes mitosis to produce all the cells in the body, right? Sperm and egg come together, make this single zygote cell that has a combination of DNA from both parents. And then that cell undergoes mitosis, which we already talked about a whole lot really fast, and grows into an organism, a person, a cat, whatever, right? Now, early in that development, so pretty early on in that little zygote dividing into a little blastula, right, a little ball of cells, a certain number of those cells, as it's dividing, get kind of set aside, right? And these cells that are kind of set aside really early in development are those that will be able to do meiosis, okay? And these are called germline cells. You have them from the time you're born. There's a chunk of cells set aside that will be for making gametes whenever, right, the stage of making gametes occurs, right? So, and those are called germline cells. Now, germline cells, like somatic cells, are diploid. The only difference is a germline cell can do meiosis, a somatic cell cannot. All right, so early in development, these germline cells get set aside. They look very much like all your other cells, but they are capable of reductive division. Right, they can undergo cell division that will produce gametes, basically. Right, so we know why meiosis. So let's do a little bit of what meiosis is, basically the rest of the chapter. So there are some features of meiosis that, one, differentiate it from mitosis, and two, are just kind of critical in the general evolution of species, right? So we're going to talk about how homologous chromosomes, right, these kind of pairs, right, both my chromosome 1s, both my chromosome 7s, whatever, are behaving in relation to each other, right? And then we'll actually see kind of why meiosis I is called reductive division. We'll see kind of mechanically what's going on. So in diploid organisms, such as ourselves, meiosis actually happens in 2 rounds, right? So watch it. So we'll call them very nicely meiosis I and then meiosis II. Each of these contains the same phases we saw when we talked about mitosis, right? Prophase, metaphase, anaphase, and telophase. And they'll look similar-ish with some differences, right? So meiosis I happens, and it involves prophase, metaphase, anaphase, telophase, and then we go into meiosis II, prophase, metaphase, anaphase, telophase again. Okay. So when we're talking about them, we'll start with prophase I, right? And so this is prophase of meiosis I, just to kind of keep them straight so we know which one we're talking about. So prophase I is the prophase phase of meiosis I. Fair enough. And so, similar to prophase and mitosis, right? All of our chromosomes have condensed down. We've gone through S phase, they're doubled up, right? So we've got these little X's all compressed down, all in our nucleus. If we're looking at them with a microscope, we can see them, so we know we're in prophase. In prophase I, this kind of particular kind of organizational step happens where each of these pairs, right? I've got 46 chromosomes, right? 2 versions of all 23 different kinds of chromosomes I can have. In prophase I of meiosis, each of those kind of similar pairs, right? Like my 2 chromosome 7s, my 2 chromosome 9s, become closely associated. They're going to kind of find each other in the nucleus, basically, and that's called synapses. They're going to stick together. They're going to line up and stick together. Right? I'm going to organize all 23 pairs, right? My maternal, my paternal version of everything. Each one is going to come together and line up. All right. They become connected by, it's synapses, so they're connected by the synaptonemal complex, which is made of a bunch of proteins that really don't care about naming, just to know that, right? So like if this is my maternal chromosome 1, there's my paternal chromosome 1, they line up basically end to end, right? Because they're carrying the same genes, just different versions of the same genes, right? Whatever my mom's hair color is, whatever my dad's hair color is, et cetera, on down. So they're lined up, and then they're connected by all these proteins. Now notice, this is important, and it kind of, I don't want to say it makes it confusing, but it just makes it, there's another thing to keep in your head, right? Because remember, we've already gone through S phase. So here's my maternal chromosome, right? It has already gone through S phase. It's already made a copy of itself, right? So it's still, so that's why, right? So we've got our little X shape. We've got copy 1 and copy 2 of my maternal chromosome 1 stuck to my copy 1 and copy 2 of my paternal chromosome 1, okay? All right, so they're stuck together. I wouldn't want this sweater on the whole time. At the synapse-enable complex, right? By some cohesin proteins. It's going to be cohesin everywhere. We're just going to stick all kinds of stuff together. At this point, and we'll kind of come back to this a couple times, recombination or crossing over occurs at chiasmata. Worry less about the word chiasmata, but focus on the words recombination or crossing over, right? Because say I have a gene, what is that one, right? You've got the gene that makes you like cilantro. I freaking love cilantro. It's right here. I inherited it from my mother. Crossing over means that when these two line up for meiosis, we can swap it, right? So that where it used to be located on this chromosome, now it's located on this one. So you kind of swap things between your maternal and paternal homologs. It's called recombination, right?
So how do you, is it like a perfect split between your parents, or is it like a 60-40?
You between your parents?
Yeah, like if that's how you.
You are a direct split, right? You got 23 copies of your dad's DNA and 23 copies, right? 23 chromosomes of your mom's DNA. This means that when you, if you have kids, the kids will not get a 50-50 split of that DNA because it's going to kind of shuffle itself. And it's actually going to shuffle itself twice. This is the first shuffle. Yeah, I know. Right, so we'll come back around. We'll come back around. All right, so these homologs through most of meiosis I are going to stay associated. The sister chromatids are going to stay associated till we get all the way into meiosis II. We'll come back to them. In anaphase I, the homologs, not the sister chromatids, right? So we got the X's, and the X's are stuck together. In anaphase I, the X's come apart and go to opposite sides, right? Remember in mitosis and anaphase, we pulled the X's in half, right? And sent 1/2 to each side. So this results in, right? My X's are stuck together. I send my maternal X over here and my paternal X over here. So each of these two new cells has one copy of each of the 23 chromosomes, right? One version, right? The cell either ended up, right? It's got 23 chromosomes in it from either my mother or my father. So it is haploid. That is the reductive division part. We just did the reductive division. We went from being diploid with 46 chromosomes to putting 23 in one cell and 23 in another cell, right? In mitosis, we had these 46 chromosomes and we just pulled them all in half, right? And we sent 46 that way and 46 this way. Maybe. We'll get there. All right. We'll go through it a couple times. So this happens, and then it's followed by Meiosis II, which basically just looks like mitosis. Meiosis II will behave almost exactly like mitosis. But we don't replicate DNA a second time, right? Because if I send my maternal chromosome 1, the whole thing into this cell, right now it's like one of those little X's that we saw in mitosis. So in meiosis, anaphase 2, we'll just pull the X in half and do the same thing we did in mitosis. We don't need to replicate again. And ultimately at the end of meiosis II, we have 4 haploid cells. Let's walk through step by step. So we're going to watch what the chromosomes are doing during this meiotic division, because it is different. Talk about monopolar attachment. It's critical. And then we'll compare what's happening in anaphase 1 and anaphase 2, because those are kind of the two big moments in meiosis. So prophase 1. In prophase 1, our homologous pairs, right? Stick with this, my maternal chromosome 1 and my paternal chromosome 2 are acting as a unit in prophase 1. So after interphase happens just like normal, right? Go through G1, we go through S phase, we replicate, right? Each chromosome kind of gets its own little copy attached to it, those little sister chromatid pairs. So my maternal chromosome 1 is an X because it's copied itself, right? Attached to its little centromere. Male, paternal, whatever, chromosome 1 has done the same thing. It's a little X shape, little pair of sister chromatids, and then they find each other. So in the nucleus, these homologous pairs attach to the nuclear envelope, right? So they're in the nucleus. And so chromosome 1 from my mother, chromosome 2 from my father find each other, and then they attach to kind of the membrane of the nucleus, which allows them to line up right next to each other, right? Which will make that kind of sticking together, synapses, a little easier, right? So they're joined along their entire length, right? And so the sister chromatids kind of along their length are joined by this meiosis-specific cohesin. That's fine, just more cohesin, don't stress, more cohesin, holding them together. All right, so prophase 1. All 23 chromosomes find their little friend chromosome, stick together, get all nice and organized. Again, this is where crossing over occurs. We've got our little sister chromatids kind of glued together. They're kind of glued together this way, right? And they're lined up. So we can start trading kind of gene for gene, right? I don't know. My dad's green eyes for my mom's blue eyes. Doesn't really work like that. That's also not what color their eyes are. Doesn't matter, right? Gets swapped, right? So where initially this copy of chromosome 1 were all genes from my mom, now it's a mix. This chromosome was initially all genes from my dad, now it is a mix. It's shuffled back and forth that way. All right. Once crossing over and recombination, either way, is finished, that synaptonyl complex breaks down. So they join up, they trade some stuff, and then they let go. All right. Metaphase 1. So they line up, similar to metaphase in mitosis, right? They go to that metaphase plate, they all go to the middle of the cell, get themselves all in the center. With the difference that when we drew this for mitosis, right, they were kind of in a single file line. So we're going to pull each of these X's in half. And meiosis, that's not what we're going to do, right? So chromosome 1, maternal and paternal, are going to come up next to each other and line up. Chromosome 2 is going to come next to each other and line up. Chromosome 3 is going to line up. Now, what side the maternal and paternal ones are on can vary. It's less important, but we have a pair of chromosome 1s, a pair of chromosome 2s, chromosome 3s, and so on. We'll line up on that metaphase plate. And then just like in mitosis, we've got these spindles with microtubules that are going to come out and grab these guys at the kinetochores, but different, right? The spindle's going to come out and it's going to grab both kinetochores of this whole chromosome, right? It's going to grab the entire X. So this entire chromosome is going to be monopolarly, that's not a word, but is going to be attached to a single pole, a monopolar attachment, right? Where in mitosis, right, we attach them at both sides and pull them apart, bipolar attachment. Now this whole chromosome is just attached here. This whole chromosome is just attached to this side. Right? So we have monopolar attachment. Each entire chromosome is attached to either one side of the cell or the other. right? And so these are, just like in mitosis, this is what's going to kind of be pulled apart into our two separate cells. Right, so metaphase 1. Anaphase 1, the microtubules shorten, right? Just the same. Breaks the chiasmata between the homologs, right? Because these guys were kind of associated with each other a little bit. In anaphase 1, we're going to pull them apart and then send this entire chromosome with both of its sister chromatids to one cell, and this entire chromosome with both of its sister chromatids to the other cell. Do that for all 46 chromosomes. So you end up with 2 new cells with 23 chromosomes each. Right, and the meiosis-specific cohesin that was holding the homologs together is destroyed, is gone. Right, compared to mitosis, right, where we had bipolar attachment, right? The single chromosome was attached to both sides. And so we're here in metaphase, and in anaphase of mitosis, we pulled this thing in half. Right, we're not pulling in half now. We're just pulling the whole X. Right, in anaphase 1. All right. So this is where shuffle number 2 happens. So the random orientation of these chromosomes on the metaphase plate, right? And it's kind of where each maternal and paternal homolog is, right? Because you could line up like this. I could end up sending all maternal chromosomes to one side and all paternal to the other side. Is it all paternal to this side and all paternal to that side? I could do some mix, right? This one gets paternal 1 and 3 and maternal 2, and this one gets paternal 2 and maternal 1 and 3, right? So each new cell gets either a maternal or paternal copy. Which one it gets is random. That is independent assortment. So the copies of each chromosome are kind of independently, right, shuffled into each of the 2 new cells. Right, and so this is where I would not pass on evenly like all my mom's genes or all my dad's genes, right? Because any gamete that is produced in meiosis is going to hold a mix of my parents' genes. Right? We'll have some of my mom's chromosomes, some of my dad's chromosomes. Hopefully one of each of the 23, but a mix of the 2 sets. Telophase, right, is just reverse prophase, and it looks roughly the same. Chromosomes, right, segregate to opposite sides. We remake the nucleus. Everybody divides, does their thing. Now the chromosomes are still pairs of sister chromatids, right? So we didn't pull them in half, connected at their centromere, but they're no longer identical, right? So we now, like you remember mitosis, we had these sister chromatids, and sister chromatids were identical. So we're making 2 identical cells. This time, because we've done crossing over, not only have I randomly shuffled my mom and dad's genes into 2 different cells, each of those chromosomes now has a mix of maternal and paternal genes because we did crossing over, right? So telophase happens, we make these 2 haploid cells, 23 chromosomes each. Each chromosome still a pair of sister chromatids, followed by a brief interphase. No S phase, right? We still have pairs of sister chromatids. We don't need to double things again. All right, so no S phase in the second interphase. And that's meiosis, the main bit, right? Looks like this. Less like maybe. Right. Prophase 1, all the chromosomes condense into the center. Each pair of chromosomes kind of finds each other, lines up. We do some genetic shuffling. Metaphase, each of those pairs line up together in the center. Anaphase, we separate the pairs, right? One whole paternal chromosome goes this way, one whole maternal chromosome goes that way, right? For each chromosome. And then telophase, we remake our nucleus, we divide them in half. We end up with 2 cells, 2 haploid cells, right? Meiosis I. Meiosis II basically just looks like mitosis, right? So we have, but it's happening to these two cells, right? So meiosis I made two cells. Here's these two cells. We now have a haploid number of chromosomes. We started with 4, now we've got 2, right? So prophase, they condense, right? Metaphase, they line up in the center, single file, just like mitosis, right? So we have now bipolar attachment, right? So these spindles are going to come in from either side, grab a kinetochore on either sister chromatid in metaphase, and then in anaphase, we're going to pull those two sister chromatids apart, right? Just like we did in mitosis, right? And then each of those cells will go through telophase, right? We'll remake our nucleus around these sister chromatids. We'll divide. We're going to have 4 haploid cells with single copies of everything. And that's meiosis II. We end up with 4 haploid cells. So let's talk about the specific differences between meiosis and mitosis in kind of a nice listy sort of way, right? We'll just talk about some specific differences between the two, right? So here's the whole thing laid out, if you like having kind of a whole figure laid out to look at, right? So we've got meiosis I and then meiosis II, right? And if you kind of look at meiosis II, it looks really similar to mitosis, just with fewer chromosomes, right? So if we have the same individual that has 4 chromosomes, right? We can take all 4 chromosomes through mitosis and create 2 new cells that still have 4 chromosomes, or take this cell and go through meiosis, go through meiosis I to end up with 2 chromosomes each, and meiosis II, right, to separate those sister chromatids apart. Right, so that's kind of just there to summarize it all for your studying purposes mostly. So 4 distinct differences between meiosis and mitosis. Maternal and paternal copies of each chromosome pair up and exchange genetic material between each other. In meiosis I, specifically anaphase I, the sister chromatids of each chromosome remain together, connected at the centromere. And so the entire, right, kind of X shape of the chromosome is moved to one or the other of 2 new cells, right? Sister chromatids remain connected at the centromere in anaphase I. They will not in anaphase II, right? The kinetochores of any, right, and the same thing, kinetochores of those sister chromatids, right, it's on either side, are attached to the same pole. That's that monopolar attachment thing, right? So here's a pair of sister chromatids for chromosome 1, pair of sister chromatids for chromosome 1 other copy. Both kinetochores of this pair of sister chromatids are going to be attached here. Both kinetochores of the sister chromatids here will be attached there, right? That idea of monopolar attachment. Right. Lastly, DNA replication will be suppressed between meiosis I and meiosis II. Right. So we'll have an S phase before meiosis I, but not before meiosis II. So if we double and divide and then double and divide, then we're not really doing reductive division, right? So we had to leave it out somewhere. Why? Why do all this complicated stuff? Because cells produced by, 1, we need reductive division. 2, there's kind of some additional benefits that come along with doing things this way, right? Bacteria don't do reductive division. They just keep dividing forever and they're fine. So what else do we get out of doing this really complicated process? So the cells produced by mitosis are identical, right? When cells in your body divide, they double up their DNA, they divide in half, and you get 2 cells that are identical to the first cell. Right? And this is critical. This is super important, right? For growing, for development, for wound healing, all done through mitosis, just dividing cells and making cells that look exactly like the cells they came from. Fine. Cells produced by meiosis are not, right? Each cell is getting a mix of maternal and paternal chromosomes or homologs, And those individual homologs, right, vary because of crossing over. So you kind of get this double shuffling of all of your genes and genetic variability available to you, right? Therefore, populations that reproduce sexually have a greater genetic variability than asexually reproducing ones, right? Because crossing over occurs, because independent assortment happens, you get just kind of a much greater amount of heterogeneity or diversity, right? Genetic diversity in populations. It leads to patterns that we can observe. And it's the basis, right? This whole process of meiosis, crossing over, independent assortment are the basis for the study of genetic heritability, which is basically all of chapters 12 and 13, right? So as we go through chapters 12 and 13, we're gonna keep referring back basically to crossing over, independent assortment, and the effects that that has on kind of the genes you inherit, the genes you pass on, the genes you express, things you don't show, right? All of that sort of stuff. All right. Now, yes, that gets us to the review. I'm not going to immediately turn around and hammer you over everything I've told you in the last half hour. Okay, I'm gonna let you go a smidge early, but I want you to go back through chapter 11, walk through it, draw it out, find a whiteboard, whatever works for you, right? Talk it out. And then when we come in on Wednesday, we'll go through and review chapter 11, make sure we're all kind of square on these ideas, the things that are happening, so that we can kind of go into chapter 12 and chapter 13 well prepared for it. All right. So have a good rest of your Monday.